Okay, so this is Chapter 5 review. Now, we didn't make it all the way through Chapter 5, so we're going to pick up the last part of Chapter 5 for Test 2. But what we covered for this is for the test. Okay, so coming back to the idea of a bilayer membrane. We talked about this before. Now we're going to start getting into the physiology of the bilayer membrane. Before we talked about what it was made up of, which is phospholipids. Okay, recall these have the two fatty acid tails, and on one they have a phosphate group, and this gives it a dual nature. One is hydrophilic, one is hydrophobic, meaning the nonpolar tails are hydrophobic. They don't like water. The polar head with the phosphate group, see that blue sphere? That's hydrophilic. It likes water. Okay, we talked about this in previous chapters. And again, we've seen this before. The head region of this likes water, so it will want to be near water. The hydrophobic tails, like a phobia, if you're an arachnophobia, you don't like spiders. Well, if you're hydrophobic, you don't like water. Okay? The tails, the nonpolar tails, will not like water, and this arranges the bilayer membrane. We talked about this before. I'm not spending a lot of time on it here. Just a kind of review for you. Okay? Now, we're going to delve into the actual physiology of this bilayer membrane. We describe the bilayer membrane movement, 3 words. Fluid mosaic model. Okay? Go ahead and star that. Put a little dot next to that. You need to know that. The fluid mosaic model describes the structure of the bilayer membrane. Couple points here. Our membrane is not a rigid structure. It is a dynamic, moving structure. Think of a waterbed, how it moves up and down. That's kind of like how our membrane is in motion. Okay? Another part of this is proteins located on the membrane can move around the membrane. Okay? Many proteins are not fixed in positions. They are movable. In fact, many times 2 proteins have to move together on the membrane to do a function. Okay, so just reiterating this, that all of these things are movable. Okay, there's motion going on our membranes. They are not locked in. And this whole idea we described in class, we said was the fluid mosaic model. Our membranes, the bilayer membrane, made up of phospholipids, is in continuous motion. Okay? And this motion is important for life. If we don't have this motion, the cell dies. Okay? Proteins don't come together. Things cannot leave and enter through the membrane. The membrane has to have some degree of motion in order for life to exist. So, couple things here with the fluid mosaic model. Conditions of the cell can change the fluidity. Okay, for example, temperature. As temperature goes up, fluidity goes up. Now you kind of can see this with good way of thinking about this is butter, right? Butter in the refrigerator, solid. Butter outside, it melts. Well, the idea is kind of a similar idea, that as the temperature becomes higher, fluidity of the membrane goes up. As temperature goes down, fluidity of the membrane goes down. Okay? Now, back to this idea of changing fluidity. The cell is not an inactive bystander here. Cells can add molecules such as cholesterol to change the fluidity of the cell. And we described cholesterol in class and what that did. And we used the idea of tropical fish versus cold weather fish as an example. You might recall that from your notes. Okay, this kind of describes the fluid mosaic model. The sphere on your left is, well, these are 2 cells, okay? And the dark green spheres and the light green spheres are proteins. And we use this to describe what would happen based on the fluid mosaic model if we combine these 2 cells together. So this is separate, combined, fused, and then overall what happens. And what do we see? That when we combine these 2 spheres, Okay, the green and the dark green and the light green proteins, because of the fluid mosaic model, because these are in motion through the membrane, you're going to get a mix of proteins along the surface, and that's all it's saying. Okay, if it was not for the fluid mosaic model, as we put these together, one side would have dark green, one side would have light green, but we don't see that. We see there's a mixing because these proteins are in motion. I think I used an example of, if you've ever been to Mardi Gras, okay, and you see that huge crowd of people, right? Think of each person as one part of the membrane. They're all moving around. Okay, that's the kind of idea you want to think about here. Okay. Now, there is a need for fluidity, but there has to be a balance. Kind of like that idea of homeostasis. Remember we talked about homeostasis being just right. If it's too less or too much, it's not good, but that just right in the middle is good. Okay? Same thing with fluidity here. The membrane can be too fluid. Okay, and if a membrane is too fluid, it's literally going to rip itself apart. It's going to form punctures and holes, and it can't maintain stability. So having too much fluidity is a bad thing. But on the other side, having too little fluidity, again, is equally bad. Okay, so there has to be a just right level of membrane fluidity. Too much is no good, too little is no good. Okay, so not enough fluidity, it's too static, that's bad. Why? Well, one thing, proteins can't move around the membrane to come together to do a function. It's hard to get things in and out of the cell if the membrane's not fluid enough. But if it's too fluid, again, equally bad. It's so fluid that you have proteins moving around, they don't come into association long enough to do something. You can get rips and tears, and you can lose membrane integrity as you get too much fluidity. Think about that picture we had moving before. Think about that just going up really high, and what would happen? Rips and tears start forming in the membrane. So the point here is that there is a certain level of fluidity the membrane must maintain. Above it, no good. Below that, no good. Okay. Now, proteins can fit between the cell membrane. So when we look at our membrane, if we could look at our membrane, we would see that it was studded with all sorts of proteins. Looking on the picture starting from the right, it says lipid bilayer, and we look over a little bit and we see these orange cylinders. Okay, those are what we call transmembrane proteins. Those fit in between the membrane. Very important. Many of these are receptors, they're channel proteins. We'll talk about some of their functions in this later on, but for now we're going to say that our membrane has proteins going through it. We call those transmembrane proteins. Sometimes they're on the surface, those are peripheral proteins. Okay, but the point here is what we have proteins in close association with our bilayer membrane. Okay, and many of these move around the bilayer. Lots of different types of proteins we're going to see on the bilayer membrane. Okay, we're going to go through each one of these in detail. This is just a summary slide of those proteins. And again, summary slide. Kind of coming back to the idea I've used before where membrane has, it's like a face, right? And I've said that it lets things in, it lets things out, right? And like it detects things about the outside. Kind of like I said that a mouth lets things in and out. Your nose and eyes detects things around the world. And it's your shape of your face lets other people recognize you. Same thing with the membrane, okay? It's like a mouth. It's going to let things in and out. It's like your nose and ears. It has receptors on it that detect things about the environment. It has tags on it, for example, MHC markers that tell other cells what type of a cell it is. Just a summary of what we talked about before. Okay. So now we're going to talk about some types of proteins that we see with the membrane. First one is adhesion proteins. These help the cell attach to the outside environment. Enzymes. We'll talk about enzymes later, but enzymes are molecules that catalyze chemical reactions. Receptors, really important one here. Remember how we said it detects things about the outside? Well, these receptors are one of those, as one way it does that. So a receptor is going to pick up signals, like from a hormone, tell the cell inside to do a particular function. Good example of this is the hormone insulin. Okay, so insulin, if you know someone with diabetes and insulin, you know there's something going on with blood sugar, right? Okay, so hormones. The hormone here is insulin. Insulin does not enter a cell. An insulin hormone is going to stay outside the cell. So think of insulin, and this hormone is this protein, and it's outside the cell. And it comes next to the cell, and it fits into the receptor, kind of like a lock and a key. Well, that receptor then goes through the membrane and inside the cell and starts a signaling pathway that leads to changes in sugar. Okay, we'll talk more about that later on, but for now we're just going to say that that receptor picks up a signal from the outside, example insulin, and it communicates that to the interior. We talked about this in depth in class, and you probably want to refer to your notes on this. Recognition proteins, these identify cells as self cells. Again, we talked about this, and we highlighted the idea of transplant patients and what kind of drugs they have to have, immunosuppressive drugs. Next are these transporters and these channels. We're going to talk about getting things through the membrane. Okay? Many times, something needs to go through the membrane, it just can't fit between the lipids. It has to go through a tunnel or a channel of sorts. And that's where these transporters come in. We will talk about this with membrane transport, but these are proteins that we see inside the membrane. Okay, so let's talk about transport across the membrane. New topic. How do we get things into and out of a cell? Right? We've said that the membrane does that. Remember, we've talked about that. It gets things in, it takes things out. But how does it do that? Before we get into that discussion, we got to give an idea of concentration gradients and what concentration is. Okay? So let's talk about concentrations and concentration gradients. Okay, so concentration is the number of molecules per unit volume of a substance or of a volume. So for example, let's use yellow dots as a molecule, and let's use these gray cylinders as a unit volume. If something has high concentrations, it's going to have a higher number of molecules per the same area. You see the gray cylinders here, they all have the same volume. The one with high concentration has more of the yellow dots, more molecules per the volume. Okay, so when I talk about high concentration, I mean that something has more molecules per unit volume than something with low concentration. Now let's combine two of these with the pathway and see what happens. So let's just say we took that high concentration cylinder and we connected it to a low concentration cylinder and kept that pathway closed. Okay? Got to ask you a question. What's going to happen here? If I connect a cylinder, a bottle, okay, with a lot of molecules in it to an equal size bottle with very few, what's going to happen? Well, what's going to happen if we open this passageway up? You tell me. Well, what's going to happen is the molecules from the high concentration are going to move to the side with lower concentration. Okay? This is diffusion. Now, they're going to move over until you have an equilibrium point where the left and the right side have about the same concentration. And that is called equilibrium. Now, what happens? I think we see what's happening here, that it stops when they each reach a medium concentration, when they both have the same concentration and they have become equilibrized. Now, they're equal in concentration. Now, what's a concentration gradient? You're going to hear this term a lot in biology. Concentration gradient, concentration gradient, concentration gradient. When you have a difference in concentrations between two regions, that is a concentration gradient. Okay? One area has a high concentration, one area has a low concentration. That's a concentration gradient. So this is a large concentration gradient where one has a lot of concentrated molecules, the other has very few. This is a medium concentration gradient. Okay? The difference between the two areas is less. And this is a small concentration gradient. Okay? And this is no concentration gradient. So diffusion is the net movement of molecules down a concentration gradient. We open that spigot up, like we said before. What's going to happen? Well, those molecules are going to move down the concentration gradient, okay? And they're going to go to the area of lower concentration. So before, what's going to happen here? Let's set this up this way. So what's this? This is a concentration gradient. High concentration on the left, low concentration on the right. We open up the tunnel between them, and the molecules have gone down their concentration gradient, and they have, therefore, undergone diffusion. Okay. So when we say diffusion, this is diffusion. The movement of molecules from areas of high concentration to areas of low concentration. That is diffusion. So why is this important? Well, we had to review this because we need to talk about the direction a molecule moves down and through the membrane. Does it go inside to outside the cell or outside to inside the cell? The discussion we just had kind of goes to answer that. So let's talk about movement across the bilayer membrane. So there are 4 ways a molecule can exit or enter a cell. We're going to talk about each one of these. 1 is through the bilayer membrane. 2 is through channels inside the bilayer membrane. 3 is through vesicles. And 4 is through a pump. We're going to talk about each one of these in detail. So let's first talk about directly through the bilayer membrane. So, do you remember before we had 2 gray cylinders? And one had a lot of yellow dots and one had very few yellow dots? Well, think of that, and one gray cylinder would be the blue layer at the top of this image. Another of the gray cylinders would be the peach color below here. Okay, so before we had 2 cylinders with different concentrations. Well, along the membrane, we have an area with more concentration of molecules, and the other side has a lower concentration of molecules. In that case, those molecules are going to move, or those atoms are going to move from high to low concentration. Okay? So, before diffusion, what happens? We have a lot of these atoms or molecules on one side and very few on the other. What will happen? Well, diffusion tells us this will happen. That some are going to move from areas of high concentration to low concentration until they form an equilibrium, until they have equaled out their concentrations. So, a couple of factors that go into this. 1 is size. The bigger they are, the slower it is to move through the membrane. The smaller it is, the faster. Another important factor here is steepness of concentration gradient. If you have a very large difference in the concentration gradient, it's going to move faster. If you have a very similar concentration, one's maybe a little bit more concentrated than the other one, it's going to move slower. Finally, charge. If that molecule or atom is charged, it's very hard to move through the bilayer membrane. Okay. If it's no charge on it, for example, steroid hormones, you're going to get faster diffusion. And we talked about this somewhat in depth in class, and you may want to review your notes on this. So some materials pass through the membrane. Some materials do not. What's going to move through the membrane? gases, for example, oxygen, carbon dioxide, and small nonpolar molecules tend to move through the membrane relatively easily. So that's what moves through the membrane. What does not move through the membrane? Well, large molecules like glucose, a lot of ions like calcium and potassium, these simply cannot move through the membrane alone. Okay? They have to have a channel. We'll talk about that coming up. So, another way you can have membrane transport is through the bilayer membrane through channels. Okay, you have protein channels. Think of like a tunnel that is going to connect the outside and the inside of the cell. So look at this picture here. This, at the bottom, those red spheres, those would be small nonpolar molecules. Okay, the green spheres, those would be larger molecules or polar molecules. Both of these, what direction are they going? They're going from high concentration to low concentration. So they're going the same down the concentration gradient. But what's the difference? Here, there's a tunnel that connects the two. And we call this facilitated diffusion because there's a channel, a tunnel, that is helping diffusion take place. Okay? So all of this is facilitated diffusion. Facilitated diffusion works with the diffusion of large molecules and charged molecules, or atoms. Okay, because these cannot simply fit between the bilayer membrane alone. They have to have a tunnel or a passageway to do it. Now we say facilitated diffusion. What's facilitate? Facilitate means to aid. If you're a facilitator, you help someone out. So facilitated diffusion is there's assistance going on with diffusion. Diffusion is what? Areas of high concentration to areas of low concentration. Facilitated diffusion is the use of a channel, a tunnel, to go from areas of high concentration to low concentration. Why is this needed? Because the objects going from high to low, because of their makeup, simply cannot fit in between the membrane because they might be too large or they might have too much of a charge. Okay? So the third way you can do membrane transport is through a vesicle. It's like a small ball in a sense. Okay? So vesicles are spheres of molecules, and we use these vesicles to bring things into the cell and to release things out of the cell. For example, some cells are going to bring in bacteria into the cell using a vesicle transport. Some cells are going to make hormones, and they're so large that they put those hormones into a vesicle and release them to the exterior. Okay, because these things are so large that they simply cannot go through the channels we talked about before. Two types of vesicle transport. We have endocytosis and exocytosis. Endo is what? Inside. Okay, so endocytosis is vesicle transport where you're bringing something to the inside. Exocytosis is the opposite. Exo, outside, like an exoskeleton. You transport things from the transport things from the inside to the outside. So down here, this is exocytosis. We have molecules in the cell, and they're in a vesicle. So the round sphere on the left of the image, that's a vesicle. That sphere fuses with the membrane, and that membrane then opens up and releases those proteins to the outside environment. That is exocytosis. Endocytosis is the opposite. Starting at the left, a tuck, invagination, a notch forms in the membrane and molecules enter it. That pinches off and forms a vesicle. This is endocytosis. Here's a little video of endocytosis. We have a bacterium, too large to bring in through a channel. So that bacterium comes in, it wraps around that bacterium, forming a vesicle, and that vesicle is then brought inside the cell. This is endocytosis. An example of exocytosis. We need to get rid of a lot of waste, okay, or this could be a hormone, could be several different things. So we have those in a vesicle, a round spherical membrane ball that fuses with the outside bilayer membrane, opens up and releases it into the environment. That is exocytosis. Finally, let's talk about using a pump. Okay, the last one we're going to talk about with membrane transport. Now, before we talked about this, we said that one on the left is simple diffusion. These are small molecules, uncharged molecules that can pass through the membrane, going from high concentration to low concentration. We talked about facilitated diffusion. We had some that were very concentrated, but they had to use a channel to go to areas of low concentration. The green spheres on the image, that's facilitated diffusion. But what happens if you want to go up the gradient? What if you want to go up the concentration gradient? What if you want to take a molecule or ion and push it toward areas of higher concentration? Well, that is active transport, and we use the pumps to do this. Okay? This process is called active transport, and it requires the input of energy. So this is sarcoplasmic endoplasmic reticulum, ATPase. Don't worry about the molecule here. Okay, we're not worried about the molecule. Let's just talk about the direction of calcium. So inside the sarcoplasmic reticulum, we have what? 4 calcium spheres. Okay, inside the cytoplasm, we have 2. And the number here is representative of the concentration. So we have more calcium in the sarcoplasmic reticulum than we have in the cytoplasm. But what if we want to get more of that calcium into the sarcoplasmic reticulum? Well, what do? We're going to use a pump. And what's going to happen is, if we wanted to go down the concentration gradient, that the calcium in the sarcoplasmic reticulum would go through it and into the cytoplasm. But we don't want to do that. We want to go the opposite way. So what do? We want to go up the concentration gradient, and we're going to use a pump to accomplish that. So we're going to put energy into this pump and watch what happens. Before we put energy into it, we had 4 calciums in the sarcoplasmic reticulum and 2 in the cytoplasm. We put energy into it, and then we get those 2 from the cytoplasm into the sarcoplasmic reticulum. Now, this is very important when we talk about membrane physiology, but for now, what you need to know is this. Active transport uses energy. Active transport moves molecules or ions up the concentration gradient from areas of low concentration, as shown in the cytoplasm, to areas of higher concentration, shown above in the sarcoplasmic reticulum. And that is active transport. Okay? It's like a bilge pump in a sense. It uses energy and it pushes ions or molecules up or against the concentration gradient.